Hi, I am Dr. R. Nirmala Devi, Assistant Professor in the Department of Biochemistry, Biotechnology and Bioinformatics, Avinashalingam Institute for Home Science and Higher Education for Women, Coimbatore. You have chosen to do the lesson on enzyme technology, introduction and production of enzymes. This module would focus on enzyme technology, which deals with the enzymes, the metabolic catalysts and their use in various industries. The introductory part outlines the concepts in enzyme technology as part of biotechnology. Briefly, it describes the methods involved in the large-scale production of enzymes through fermentation process. The objectives set for this module is to gain knowledge about enzyme technology and production of enzymes and to provide an overview of enzyme production processes. Basic knowledge about the enzymes and their chemistry is of prime importance. This knowledge, together with state-of-the-art technologies with protein engineering, is the basis for discovering and improving new enzyme applications. The learner will be able to understand the concepts in enzyme technology and methods for the production of sustainable and high value-added products utilizing enzymes as biocatalysts and microbes as efficient producers in order to meet various human needs. Let us first take up a brief introduction on enzyme technology. Enzymes are now largely used in a wide range of industrial processes. The study of industrial enzymes and their uses is called enzyme technology. The goal of enzyme technology is to design innovative products and processes that are not only competitive but also meet the criteria of sustainability. Proteins are the metabolic end products of gene expression and they characterize different organisms. They may be either structural or functional proteins. Enzymes are the functional proteins which catalyze biochemical reactions required for the survival and reproduction of the living organisms. In some cases, biocatalysts may be nucleic acid in nature as exemplified by ribozymes. Enzymes display a number of remarkable properties, especially when compared with other types of catalysts. The most important ones are high catalytic power, specificity and the extent to which their catalytic activity can be regulated by a variety of naturally occurring compounds. These properties of enzyme molecules lured many researchers to develop new enzyme applications and also in improving the existing applications. Although enzyme preparations have been used by mankind over a long period, a breakthrough is needed to extend their uses in broader areas with more superior performance. Recent advancements in novel enzyme exploring and engineering are briefly introduced in this module. Enzyme technology is concerned with the application of enzymes as tools of industry, agriculture and medicine. Also, with the development of modern natural science, a wide range of enzymes have become available on a large scale and this has increased the scope of enzyme technology. The research in this area includes fundamental and applied enzymology, biocatalysis, molecular modeling, structural biology and diagnostics. The overall goal is to develop new and more sustainable products, processes and services to meet the human needs or to improve processes to produce existing products from new raw materials and biomass. Enzymes can be used as biocatalysts to catalyze chemical reactions on an industrial scale in a more sustainable manner. Their application covers the production of desired products such as food, animal feed, pharmaceuticals, fine and bulk chemicals for all human material needs, as well as in a wide range of analytical purposes, especially in diagnostics. And for environmental protection, enzymes are biodegradable and unlike many inorganic catalysts, cause less damage to the environment. In the field of biotechnology, there are many industrial applications that result in biotech products which we use every day at home. Enzymes have been used in brewing, baking and alcohol production since prehistoric times. Only our ancestors did not call them enzymes, but they certainly used them. One of the earliest written references to enzymes is found in Homer's Greek epic poems dating from about 800 BC, where it is mentioned about the use of enzymes in the production of cheese. Enzymes are responsible for fermentation of sugar to ethanol by yeast, a reaction that forms the basis of beer and wine manufacturing. Enzymes oxidize ethanol to acetic acid. This reaction has been used in vinegar production for thousands of years. Similar microbial enzyme reactions of acid-forming bacteria and yeast are responsible for aroma production in bread making. Presently, more than 2000 different enzymes have been isolated and characterized. More than 75% of industrial enzymes are hydrolyses in their nature. Enzyme stabilization is one of the most important properties in basic and applied enzymology. In basic enzymology, it is of particular reference to understand enzyme stabilization principles, first elucidating how and why the enzymes lose their biological activity, and then deriving structure stability relationships existing in enzymes. In applied enzymology, the most significant goal is to achieve useful compounds by biocatalysis. Enzymes are good catalysts in terms of efficiency, specificity and regulation, are also more versatile in nature with the ability to function under mild conditions. However, they are not always ideal catalysts for practical applications because they are generally unstable and they inactivate rapidly through several mechanisms. In order to enhance enzyme stability, many strategies have been pursued in recent years. The advantages and disadvantages of using enzymes are directly related to their properties. First, we shall see the advantages of enzymes. They are highly specific, rapid and efficient in their action and are therefore less likely to produce undesired byproducts. Enzymes are biodegradable and therefore cause less environmental pollution. They operate in mild conditions, that is low temperatures, neutral pH and normal atmospheric pressure, which consequently results in low energy consumption, thereby helps in saving energy. Let us see the disadvantages now. They are highly sensitive to changes in physical and chemical conditions surrounding them. The enzyme substrate mixture must be uncontaminated with other substances that might affect the reaction. They are easily denatured by even a very small increase in temperature and are highly susceptible to toxic chemicals and changes in pH. Therefore, the conditions in which they operate must be tightly controlled. We shall now discuss the various sources of enzymes. Since time immemorial, natural enzymes made by microorganisms have been used to make products such as cheese, bread, wine and beer. Biologically active enzymes may be extracted from any living organisms. Microbes are still the most common source of industrial enzymes. Naturally occurring microorganisms are the most successful producers of enzymes, but several enzymes are obtained from renewable animal and plant sources. This knowledge has been exploited by industry for more than 50 years. Microbial enzymes are isolated from a variety of sources, and these include bacteria, fungi and yeast cells. These are the few enzymes that are isolated from bacterial and fungal sources. Bacteria and fungi are the microorganisms best suited for the industrial production of enzymes. They are easy to handle, can be grown in huge tanks without light, and have a very high growth rate. Among the many different enzymes being used industrially, over a half is derived from fungal sources and nearly 1/3 is from bacteria, with the remainder from animal and plant resources. Microbes are preferred to plants and animals as sources of enzymes because they are generally cheaper to produce. Their enzyme contents are more predictable and controllable. Plant and animal tissues contain more potentially harmful materials than microbes. For instance, the presence of phenolic compounds in plants. In addition, microbial enzymes are widely used in industrial processes due to their large productivity, chemical stability, environmental protection, plasticity, and vast availability. The microorganisms may have specific genes introduced into their DNA through genetic engineering so that they produce enzymes naturally made by other organisms. Most of the industrial enzymes are produced by a relatively few microbial hosts like Aspergillus and Trichoderma species and Streptomyces and Bacillus species. Certain industrially used microorganisms have been genetically modified to increase productivity, the desired activity, and not to produce undesirable side effects. Often, enzymes do not have the desired properties for an industrial application. Protein engineering is used to improve commercially available enzyme to a better industrial catalyst. Several enzymes have already been engineered to function better in industrial processes. These include proteinases, lipases, cellulases, and few amylases. Let us now consider the types of enzymes. Organisms produce enzymes inside their cells called intracellular enzymes and may also produce enzymes that are secreted into the medium and function outside the cells called extracellular enzymes. Now let us see the comparison between intracellular and extracellular enzymes during enzyme production. Isolation of intracellular enzymes is difficult when compared to extracellular enzymes. Cells need to be broken for the release of intracellular enzymes and is not required for extracellular enzymes. Extracellular enzymes are more stable when compared to intracellular enzymes. Separation from cell debris, other enzymes, and unwanted chemicals is needed in the case of intracellular enzymes and which is not needed for extracellular enzymes. Purification procedures and downstream processing is difficult and expensive for intracellular enzymes when compared to extracellular enzymes. We shall now discuss the methods adopted for the production of enzymes. With the recent advent of biotechnology, there has been a growing interest and demand for enzymes with novel properties. Production of enzymes by traditional mode depends on the natural host as raw materials. However, enzyme engineering has now given an option of selected production hosts such as genetically engineered microbes through mutation and transgenic plants for producing enzymes at an elevated and safe level. Considerable efforts have been devoted to the selection of microorganisms via sophisticated screening techniques and process methodology for the production of enzymes with new physiological or physical properties and tolerance to extreme conditions used in the industrial processes. The activity of an enzyme produced from a natural environment is not often favorable and satisfies the human need, and thus the activity has to be raised for industrial production. An elevated level of enzyme action is achieved by the process of mutation. An alternative strategy that has gained advantage includes the production of enzyme in a desirable recombinant organism whose growth conditions are well standardized and GRAS status is also established. The organism producing the enzymes should have a GRAS status, which means that it is generally regarded as safe. This is especially important when the enzyme produced by the organism is used in food processes. The organism should be able to produce high amount of the desired enzyme in a reasonable time frame. The microorganisms selected are usually cultured in large fermentation chambers as shown in this picture under controlled conditions to maximize enzyme production. Two different methods used for enzyme production includes surface and submerged techniques. In surface method, enzymes are produced on the surface of a solid medium, and in submerged method the microbes producing the enzyme is grown throughout the liquid medium. Both solid state and submerged fermentation methods are applicable, while the latter is preferred due to better handling of aseptic conditions, more enzyme yield and process control. The maximum enzyme production is usually in the stationary phase of microbial growth, so a batch or fed batch process is usually used. The medium must be chosen to stimulate the microbe into synthesizing the correct enzyme. For example, to stimulate a microbe to synthesize amylase enzymes, a medium with starch but no sugars is used. Downstream processing of enzyme from the raw material constitutes the key stage in the production process and uses a variety of techniques. In the first stage, cells need to be separated from the liquid part of the suspension. Separation can be done by sedimentation, centrifugation or filtration. If the cells themselves are the desired product, as in the case of single cell protein production for animal feed, then they need to be sterilized, washed, dried and packed. If the desired product is a chemical within the cells, then the cells have to be broken apart to release the chemical and the cellular components removed. The desired chemical is then extracted and purified by a number of techniques such as precipitation and chromatography. Finally, the purified chemical has to be dried and packed in a suitable form. This picture schematically represents the various steps involved in the production of enzymes from different enzyme sources such as microbes, plants and animals. The large-scale production of enzymes involves culturing microorganisms in chambers called fermenters or bioreactors. Microorganisms are best suited for use in the large-scale production of enzymes in fermenters because they have rapid growth rates and are able to produce larger number of enzyme molecules per body mass than many other organisms. Microorganisms can be genetically engineered to improve the strain and enhance yields. Microorganisms are found in a wide variety of different habitats such that their enzymes are able to function across a range of temperatures and pH. Microorganisms have simple growth requirements and these can be precisely controlled within the fermenter. Microorganisms can even utilize waste products such as agricultural waste as substrates. Coming to the requirements for the production of microbes in fermenters, oxygen is needed for the respiration of microorganisms if aerobic in nature. Otherwise, for strict anaerobes, oxygen must be excluded. A source of carbohydrate is required for respiration to release energy needed for the growth of microbes. A source of nitrogen is essentially needed in the form of ammonia and urea for protein synthesis. Both are widely used as cheaper source of nitrogen for the industrial production of microbes. Let us move on to the production steps. The large stainless steel cavity in the fermenter is filled with a sterile nutrient solution, which is then inoculated with a pure culture of the carefully selected microbe depending on the requirement. Paddles rotate the mixture to ensure the suspension is mixed well. As the nutrients are used up, fresh nutrients can be added. Probes monitor the mixture, and changes in pH, oxygen concentration and temperature are computer controlled. A water jacket surrounding the fermenter contains fast flowing cold water to cool the fermenter. Since fermentation is a heat generating process, most of the air, including carbon dioxide and other gases produced by cell metabolism, leave the fermenter by an exhaust pipe. The stainless steel fermenter with complicated control systems is not actually the most expensive part of the process. Almost 80% of the cost is accounted for by downstream processing, the isolation, extraction and purification of the product at the end of the culture in the fermenter. Isolation of enzymes. Generally, isolated enzymes are efficiently used than whole cells because enzyme concentration is higher and no unwanted enzymes are present. Pure enzymes are needed for commercial use. Therefore, microbes must be grown in aseptic conditions, free from contaminants, undesired chemicals and other microbes. It is necessary to prevent contamination with other bacteria since there may be competition for nutrients. The required enzyme may not be produced as readily. The end product may be contaminated and unsafe. The required enzyme that is finally produced must be isolated from the microbial cells. Extracellular enzymes are present in the culture outside the microbial cells since they have been secreted. They are often soluble in water, so they can be readily extracted from the culture medium and purified. These are less common in nature, although genetic engineering can be used to modify cells to promote this. These enzymes are cheaper to produce and tend to be more stable. They are therefore the preferred choice when available. Microorganisms such as bacteria and fungi are saprobionts, that is, they feed saprophytically, secreting enzymes onto their food, making them a good source of extracellular enzymes. For example, the fungus Aspergillus niger produces an enzyme called pectinase, which breaks down pectin, a substance found in the cell walls of plant cells. The fruit juice industry uses pectin widely, since when fruit is crushed to extract the juice, pectin prevents some being released and also makes the juice cloudy. To obtain an intracellular enzyme, the microbial cells are harvested either by filtration or by centrifugation from the culture and are then broken up. The mixture is then centrifuged to remove large cell fragments, and the enzymes are precipitated from solution by a salt or alcohol. Purification of enzymes. The required enzyme must then be purified by techniques such as electrophoresis or column chromatography. The desired level of purification depends on the ultimate application of the enzyme product. The industrial bulk enzymes are relatively crude formulations, while specialty enzymes undergo a thorough step of purification to yield a homogeneous product. This process is complicated and expensive, so these intracellular enzymes are used only when no other alternative is available. By their very nature, they tend to be more sensitive to their operating conditions, which make their commercial use less easy. On the other hand, these enzymes are much more common in nature. In the case of the enzymes in biological washing powders, which means coating the granules with wax to ensure that they remain dry until used. Otherwise, the enzymes would digest themselves. Thus, large-scale production of enzymes has to comply with the standards set by International Organization of Standardization for ensuring quality and production efficiency, and also environmental management control whenever applicable. To conclude, enzymes are applied in various fields including technical use, food manufacturing, animal nutrition, cosmetics, medication, and as tools for research and development. At present, thousands of enzymes are known, and of these, approximately 200 microbial enzyme types are used commercially. However, only very few enzymes are produced on truly industrial scale. With the improved understanding of the enzyme production biochemistry, fermentation process and recovery methods, an increasing number of industrial enzymes can be foreseeable. Thank you.